Okay, so we're on the very last part, part 3, and we're going to talk the Krebs bicycle pathway, and we're also going to talk a little bit about degradation of certain amino acids. So if you remember from the last video, right, we talked about there being different products from the urea cycle, right? And so remember, the urea cycle is going to make a product called fumarate, right? So urea cycle and the citrate cycle are actually metabolically linked through fumarate. So fumarate is really, really important because it's a shared intermediate. The intermediate between urea cycle and Krebs, right? Okay, Krebs cycle. So we want to be able to reuse, to continue, if we make fumarate, to then use fumarate and then be able to keep going in our urea cycle. Otherwise, it's going to back up. So what's going to happen is that the Krebs bicycle will convert fumarate into malate. Now, once we have malate, we can convert malate into oxaloacetate, and we can go through the citrate cycle. Citrate, right? So that's why they call it the Krebs bicycle Krebs citrate, right? Because you can convert it into oxaloacetate and then use it. So now we have this connection between urea cycle and the citrate cycle. And if you remember, if you're going through even part of the citrate cycle, you're going to make some energy carrying molecules that can then go through the electron transport chain and help to offset the amount of energy that's required to run the urea cycle. So, right, remember, on the left-hand side over here is our urea cycle, on the right-hand side is our citrate cycle. So what we have is the pathway in the middle that is going to link the two. So remember, we said that we make fumarate as a product, right, of the urea cycle. So fumarase will convert fumarate to malate. That's cytosolic malate. Cytosolic malate is pumped into the mitochondria. So now we have malate in the mitochondrial matrix, which is where we need the citrate cycle to occur. Because remember, the part of the urea cycle where we make fumarate is in the cytosol. So we have malate in the mitochondrial matrix. Malate gets converted to oxaloacetate as a part of the citrate cycle. And when that happens, we're actually going to convert NAD plus to NADH. Then the NADH is going to go to the electron transport chain. It's going to go and it's going to make 2.5 ATP, right? And so that's going to be great because it's going to help us offset the energy that's required, right? So the other thing that's important to remember is that there are actually 2 places where nitrogen enters the urea cycle. And one of those places is at the deamination over here, right, of our amino acids, glutamine and glutamate, right? But the other place is where we have aspartate over here, right? So we have amino acid degradation, and that degradation we convert to glutamate. We can harvest the carbon skeleton by removing that nitrate group, making alpha ketoglutarate, and having aspartate be produced. And so this conversion, right, once we have oxaloacetate from the malate, we can actually convert that into aspartate. Then the aspartate can go in. Remember, we needed aspartate to replace the AMP on the citrulline to make arginosuccinate. And that was where our second nitrogen came in. So the 2 pathways are super linked and really, really important to keep each other going. Okay, so the carbon backbones of 11 of the 20 amino acids can be converted into either pyruvate or acetyl-CoA. And that allows them to be used for energy conversion by the citrate cycle and oxidative phosphorylation reactions. So the other 9 are converted to citrate cycle intermediates like alpha ketoglutarate, fumarate, succinyl-CoA, oxaloacetate, right? But then you got to think the oxaloacetate can also be used for glucose synthesis, right, to phosphoenol pyruvate. But the amino acid degradation pathways are really, really complex. So it's much easier if we group them by what metabolites they produce. What do they make? Do they make glucose? Do they make ketone bodies, right? That kind of stuff. And so that's how we're going to talk about them. You don't have to know the big pathways. You just kind of have to understand the groups. So group 1 is going to be used to make pyruvate. We have a big group of amino acids that are made that are used to make pyruvate. And so we call these, because we're making pyruvate, we're going to call them the glucogenic, G-E-N, glucogenic amino acids, right? Then group 2 we're going to convert our group 2 to glutamate, U-T-A-M, glutamate to alpha keto glutarate, right? And so those are also going to be glucogenic. And then we have group 3, and group 3, it's not only phenylalanine, but phenylalanine is really, really important because phenylalanine actually gets converted to tyrosine, which is the only way that we get tyrosine. And then it's converted to acetyl acetyl-CoA, right? And so that makes this ketogenic, right? Because we can make ketone bodies. But the thing that's kind of important is that there are some that overlap, like threonine and tryptophan are also ketogenic, right? Because we can convert those to acetyl CoA, right? And then we can go from acetyl-CoA to acetyl-CoA, and we can make ketone bodies. So this accounts for about 13 out of the 20 amino acids. And these are the ones that you should kind of know their groups. So if you look here, if you just look at the blues and the reds, the blues are going to be your glucogenic degradation pathways, and the reds are your ketogenic degradation pathways. And if you have an amino acid where you see a bold, right, anywhere where you see bold, what you know is they're both ketogenic and glucogenic. So our first group up here, group 1, right here, these are all going to be converted to pyruvate. And then once we have pyruvate, pyruvate can go one of two ways, right? It can go to oxaloacetate, or it can go to acetyl-CoA. And once we have acetyl-CoA, we can go to ketone bodies, or we can go through the citrate cycle. The next group, group 2, down here at the bottom, these are glucogenic. These get converted to alpha keto glutarate, again, entering the citrate cycle. And then we have our group 3, and our group 3 are converted to acetyl-CoA, which can then undergo ketogenesis. So just understanding the big pathways of what the different groups of amino acids can do. So if you look, you don't have to memorize the pathways, but just kind of understand what's going on, right? Okay, so if you look at, let's take alanine, for example, right? Here's our simplest one. We have one reaction, we take alanine, we go to pyruvate, and at the same time we convert alpha keto glutarate to glutamate, and that's where our nitrogen containing group is, right there in glutamate. The carbon skeleton is preserved in the pyruvate. So really the conversion from alanine to pyruvate is a simple deamination step. Every other amino acid is going to take more steps in order to make it happen, right? So take, for example, glycine. Glycine can actually be degraded 3 different ways. Glycine can be degraded straight into serine, then serine deaminated to pyruvate, or glycine can actually be converted to a point where it's degraded completely into carbon dioxide and ammonia. So this is really actually the predominant fate of glycine in animals right here. But the other ones can happen. And the third one is to glycoxylate, but that doesn't happen very often. So if we kind of scroll up here, right, you got to think about what are kind of the products of what you're going to make. Well, if we make pyruvate, those are the blue ones that are going to be glucogenic. But these red ones up here, tryptophan and threonine, remember how we said they can go either way. So if these 2 red ones are converted to oxaloacetate or acetyl-CoA, then they can go to, we'll do them this way, they can go to ketone bodies. So it just depends on what part of the pathway they take, where they're going to go, right? And how many steps it's going to take them to get there. All right. Our group 2, these are all converted to glutamate. So all of these are converted to glutamate, and then we are deaminated. Glutamate is deaminated by glutamate dehydrogenase, and we make alpha ketoglutarate. So these are a pretty easy group, and no, you don't have to know all the individual steps. You just actually only have to know the glutamate dehydrogenase reaction. That's the deamination reaction. That one you do need to know. Group 3 is really cool. So phenylalanine is converted first to tyrosine. So this is the primary reaction that's responsible for tyrosine production in animals. And tyrosine is a very, very important amino acid because it goes on to make neurotransmitters like dopamine, goes on to make melanins, goes on to make epinephrine, all things that are super, super important. But if you have lots of it, right, then you can go on and convert tyrosine to another substrate and eventually turn it into acetyl-CoA. So that's the way that you can get rid of it. Animals can't make phenylalanine, so we have to ingest it, right? And so the first time that there was discovered a metabolic pathway that was linked to a human disease that resulted from a single gene mutation was this disease right here. And I know I'm going to pronounce it wrong, but Alcaptainuria. So this one is the enzyme that's involved in degradation of whether it's phenylalanine or tyrosine, does not matter. If you have a defect in this one, actually what's going to happen is that the substrate here, this intermediate, is going to get oxidized and then released in the urine, and it actually turns the urine black. And so you have this really cool visualization. It also leads to arthritis and that kind of stuff, but it's not terrible. But the second disorder is actually Phenylketonuria Pku. You might have heard of this one before. So this one is a defect in this enzyme, phenylalanine hydroxylase. And so this one results it's not a problem so much that, that you don't get tyrosine because you could just eat it, but it's that you have a buildup of phenylalanine. So if you have a buildup in phenylalanine, you get like 30 to 50 times the amount of phenylalanine you're supposed to get. And then this will convert it into other metabolites, and those other metabolites cause symptoms. So if you look, right, so this is what happens. We have phenylalanine, and if we can't convert it to tyrosine, this reaction does not happen. We don't make tyrosine. We could eat it, right? It's dietary, we could eat it, so that's not a big deal. But what happens is that phenylalanine gets converted into all of these other metabolites. You don't have to memorize them, but these are actually really, really dangerous and can result in severe mental retardation, stunted growth, dental problems. This was a lot of patients who were found in mental hospitals many, many years ago were found to have this. And so the primary treatment here is just don't eat a whole lot of phenylalanine. And so you'll see this on the back of food labels, PKU warning, right? Like aspartame has it. So if you don't eat it, then you don't have to metabolize it, and it's not so bad. All right, so that is it for the videos. Hope y'all have a great day.